All right, so lesson 4, HL lesson 4, but our first lesson for the HL content of cell respiration. I've got this set up. I just sent some screenshots through for what we are doing in the next week. So today's lesson is found in the week 4 section, just here, okay? So we'll do mitochondria and redox today, and then the rest are all found under week 5. We will just keep using the same PowerPoint and this same file with the questions. For this topic, I cannot stress enough, you should have something nearby that you are writing with or drawing with or putting like information down on paper, because there's going to be a lot of stuff that we're gonna talk about, and I've like specifically broken this into these 5 chunks. So please make sure as we're going, like I will say like write this down, or I will say like draw this thing. So for example, today we're gonna draw a mitochondria. I mean, if you're not ready today, that's okay. Like just go back and watch it later. But for future lessons, there's a lot of reactions, a lot of like different names of compounds. It's important to get familiar with them, with how they look and how to write them correctly. So yeah, so you'll see here I've given which slides you're going to use and what questions you can do afterwards. So today when you're done, you'll do questions 1 to 4, and you'll notice that, sorry, 1 to 5, and then after that you'll notice there's not as many questions for each one. For example, 6 to 8, only here's only question 9. Essentially, question 9 is like the notes you would take while we're going through it. Here, question 10, and then a few more to end the topic. Okay, so do take a look there and get that PowerPoint and get that document for yourself, so you're ready to go. I will spend quite a bit of time drawing. So if you want to have like the PPT on the side, that might be helpful as we're going through, because for me it's tough to switch between iPad and PowerPoint, but you can have both open at the same time. Right, so let's begin. We have been saying this a lot, like for the last 2 weeks or so, about how we're going to really dive into the arrow, right? Dive into how does the glucose actually end up as CO2 and H2O? What goes on in that reaction arrow? So all of this is about what we did in the SL lesson, which is how do we get those 36 ATPs out of the glucose? How do we change glucose? How do we break it apart? How do we do these alterations to glucose? What is the pathway that glucose takes to becoming CO2 and water? And how do we pull little parts away from it? And at the end of all of that, how do we get all this ATP? Okay, please, I must stress also, if you have a question, don't let me ramble on, because it will be rambling. It will feel like rambling. So if you've got something going on, please either raise your hand or just talk like Michael just did. I'll stop and answer any questions you need right away. Michael, is that a real question or you're just testing it out? Okay. All right, so from the beginning, what we've mentioned is we know that the energy is found in the bonds of the glucose, right? Glucose is your hexagonal shaped molecule. We're gonna see how can we like split that open and take the energy out of those bonds and use it to generate ATP. That is the whole concept of this topic. I'm gonna skip this because you can take a read through those understandings, but you will realize that you have no idea what any of it means, and there's a lot of crazy words there. This is part of the reason why we go very, very slowly through this, because there's a lot here. Okay, so to start very simply, recap from SL lesson, okay? First, your reaction, okay? glucose plus 6O2 gives 6CO2 plus 6H2O. And in that process, we are generating ATP. Now, this whole HL topic is assuming aerobic respiration. So we're gonna go, so from SL we saw glucose breaks into 2 pyruvates and then goes to the mitochondria, and then we just like skipped a bunch of stuff and said, blah, 36 ATP comes out. Well, that blah is what we will do for 5 lessons, okay? What goes on inside the mitochondria when there is oxygen available in order to get that huge ATP gain. So this is what I've been talking about. We take this arrow and we're gonna blow it up into these 5 steps, okay? We started glycolysis already a little bit. We'll go dive into that a little bit deeper. We'll do a lesson on link, a lesson on Krebs cycle, and a lesson that does these last 2 all together. Question?
Krebs cycle sounds like crab cycle.
Crab cycle?
Crab.
You might call it that. Once you get to it, you might have that feeling. Okay. The name that we give to this whole process of what happens in this arrow is called oxidative phosphorylation. Very simple. Today we'll explain what oxidation is, but I don't think this comes up later. Phosphorylation really just means adding a phosphate-related molecule. So why is this process called phosphorylation? Because we're taking this and we're gonna add it onto here, right? Making our ATP. We're gonna do that by oxidizing the glucose molecule. Now, if you don't know what oxidizing means, that's okay, because that's what we're doing today. Okay, so this term, oxidative phosphorylation, is really the term we give to it. It's like the chemistry term for what really happens in respiration. We're going to oxidize glucose in order to shove a bunch of these phosphates onto ADP. That's the phosphorylation part.
Why do they have to make so complicated names?
Sorry, I didn't hear that.
Why do they just have to make so complicated names for simple stuff? Just call it phosphate addition.
Because science. Because then you wouldn't be able to be smarter than other people. Then everyone would know anything, and what's the fun in that?
You want to make yourself more educated.
Of course. What do you think this world is all about? It's all about just like, oh, let's make this complicated so half of the people.
Oh, I'm smarter than you.
Yeah, totally. HL, SL, like IB versus whatever. Oh, you didn't take IB bio? Ugh.
Oh my God, don't talk to me.
Oh my God, you just took like regular high school bio? Yeah. Okay, moving on. This is still repeat from SL. So again, we did this section. We know we get net gain of 2 ATPs, but we're going to figure out how do we get this huge, this huge 36 when we get into the mitochondria. So to make the beginning of this easy, we're just going to draw a mitochondria. Okay, so let's swap to iPad. And if you are, oh no. If you are, oh no, what's going on? We're good. Yeah. Get out of here. Please tell me you can't see that. Okay.
See what?
Nothing, never mind. Okay, so where's my drawing? Oh, here we go. My pen. So we did draw some mitochondria earlier, like when we did the cell. Let's do a new one. Right. So the funny thing is I can't see that on my iPad. So straightforward, we'll do our outline. I'll do this a little bit off to the side so I can label it properly. Let's do red because for some reason I always associate red with mitochondria. Okay, so our classic jelly bean shape, right? Follow the IGCSE rules, no overlapping. And then we have our internal structure, which has these curvy. Yo, do you know how much easier it is to draw on an iPad? Oops, I don't like that last part. Uh-huh. That's nice. All right. So this is your basic structure. What do we need to add? We're going to add a couple little dots here. Bam. And that is your mitochondria. Okay. Why does this? Now I'm being a perfectionist. All right, good enough. All right, so labeling. As with many things in biology, structure and function, right? The structure of what we're looking at usually has some importance to the job that it needs to do. So what we're going to see is like we've been drawing this squiggly line stuff before, but we've never really explained why it's important. Okay, this topic we will get to see why it's really important that it has this shape. So first and foremost, let's be very simple. What is this outer covering called? Anyone have a guess?
Oh, it's a double cell membrane, right?
It's a membrane, right? So it is a membrane. So we're going to call this the outer membrane.
Wow, now it becomes simple. It was called phospholipid.
Yeah, false sense of comfort?
So it's actually called outer membrane.
Yeah, outer, you could say outer mitochondrial membrane if you want, actually, to be more, smarter.
Wait, actually, it's called outer membrane.
Yeah, outer membrane, outer mitochondrial membrane if you want.
Something that. Oh wow. Okay.
Yeah, and then okay, so use logic. What is this called?
Inner membrane.
No, it's called phospholipid membrane.
Big brains.
Inner membrane. And yes, so what you guys are saying actually, so if you like each of these is in fact a phospholipid bilayer, right?
Mm-hmm.
You mean like with the outer in
Yeah, both of them are their own membrane.
Okay.
Yeah.
So there's the inner and outer are separate.
Correct. So that is the same as this would also have the same thing.
You're saying okay.
Yeah, so they are both phospholipid bilayers, which again will become important soon. Okay, then what should we label next? Let's continue with logic. Okay, ready? In grade 12, so you can feel free to chime in. If you have an outer membrane and an inner membrane, what do you call the space between the two membranes?
Intermembrane space.
Wow, they're smart.
What do you mean? Wow. What do you mean? Wow?
They're not smart. They just have me.
Yeah.
Intermembrane space. That was kind of an insult, but they didn't really pick up on it. Okay, the space between the membranes. Intermembrane space. I'm gonna put a little
Is it
star.
like the cytoplasm?
No,
Okay.
not really.
What is it in that?
A star here because this is actually really important. So the equivalent, what I would say is the equivalent of the cytoplasm is inside the inner membrane. Okay, has a very cool name.
Matrix.
The matrix, yes. If you
Oh,
want,
there's an error in the matrix.
if you wanted to just be like more, you can call it the mitochondrial matrix. All of these can be, you can add the word mitochondrial, right? Mitochondrial, outer mitochondrial membrane, inner mitochondrial membrane, mitochondrial intermembrane space. Any of this can be included. Uh-oh, didn't give myself a lot of room here. Okay. The next one is this fold. Okay. The fold, oh, I shouldn't show that as being part of the outer membrane. So all of these folds of the inner membrane, okay, they're sort of like, if you imagine like villi of the small intestine, right? These are called cristae. Cristae. So all of these, this is a cristae, this is a cristae, this is a cristae, 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 cristae. Okay? Every fold is a cristae. So there are many, many cristae as part of the inner membrane. I think we only have one more label to do. And that is this. What's that?
70S mitochondria, ribosome, sorry.
Correct. 70S ribosome. Somebody tell me why or what, why is it important that this is a 70S ribosome, or what does it help to prove?
Endosymbiotic theory.
Yeah, endosymbiotic theory. What is the evidence, Gina, or what evidence does it help with?
Because 70S ribosome is for prokaryote.
Got it, right? Prokaryotes have 70S ribosomes, eukaryotes have 80S, so this proves that theory. Remember that once upon a time, one cell consumed the other cell and then didn't digest it by accident, and then somehow they ended up working together, which maybe tells us that the mitochondria once upon a time was just like, kind of like a bacteria that now is a part of all eukaryotic cells, providing it with energy. Okay, the only other label that you might see sometimes, I'm gonna switch back to our, everyone okay with the drawing? I think so, yeah. I'm gonna switch back to the PPT for a second. Did that work? You guys can see the PPT now?
看不到。
It's
No.
not the drawing.
It's still the drawing, okay. Dun, 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 dun. Oh my.
Jay yawn in there.
Hey, calm. That timing though. All right, so here we are. We're good now? Can see the presentation?
Yep.
Okay.
Mhm.
All right, so there you go. Matrix. This is two different images to show one as an electron micrograph and one as just like the drawing that you probably just did. Okay, get out of here. There we go. Can't make this there. So outer membrane, inner membrane, cristae, intermembrane space, and 70S ribosome. The one thing down here you'll see that we didn't label is the naked loops of DNA, okay? Because you can't usually, I mean, you can't really see them even in an electron micrograph. So we don't usually draw them in our drawing, but we should know that they're there because mitochondria also have their own DNA, which is also proof of what Gina just said about the endosymbiotic theory, that they have their own DNA that they make their own copies of before dividing separate from your cell nucleus. Now, one thing that I'll mention now is we're not going to, you should be able to not only label the mitochondria, but also annotate. Now, the problem with annotating is right now, we don't know all the processes, right? So we can't say like, oh, the matrix does this and the cristae does this, because if we do that, we're gonna be covering a ton of different things that we don't know yet. So what we'll actually do is at the end of everything, we'll come back to the mitochondria and you'll be able to add the like, what does each of these areas do in the cell respiration process, okay? So for now, we're just labeling, but you should eventually be able to annotate the mitochondria with what each part does. Okay, so you got a whole bunch of different versions of this drawing here. Feel free to check those out. Okay, this is what it looks like in real life. This spelling is wrong. It's not someone named Chris, okay? It's C-R-I-S-T-A-E. I'm gonna just...
Wait, what does the cristae do again?
For now, all we need to know is it's just the folds, okay? So it's just like anything else. So what does folding do in all of biology?
Increase surface area.
Yeah, that's it, right? So the folding increases surface area. We're gonna see that because it has lots of folds, it makes certain parts of respiration much more efficient by having more like membrane in order for something to take place. I don't want to get ahead of ourselves, but.
Mr. Nealon, is the black thing the phospholipid bilayer?
The black thing.
On the cristae. Like
well, the cristae. Sorry, go ahead.
Yeah, because like, you know, like the, what do you call it outside this, the nucleus, the rough endoplasmic reticulum, there's also like black things, which is the ribosomes. So is it like, does it function like the thing or no? Or is it just the phospholipid bilayer I'm just looking at?
It is really a phospholipid bilayer. Yeah, let's leave it at that, because there are like proteins and stuff embedded in here. In here, so this is membrane, right? Oh gosh, we're going off track. So this is all membrane, right? Mm-hmm. And there would be, actually it's like double membrane, like, or the phospholipid bilayer. Yeah. And then so inside of this is separate, like this area inside here is separate from the matrix out here.
Okay.
And out here. Yeah?
Mm-hmm.
I think if we check it out here. So this shows us, so the yellow is a phospholipid bilayer, right? So is this orange part.
Mm-hmm.
And then it's essentially what it's doing is separating the matrix, which is this blue, from these like sort of empty space between the outer and inner layer, the intermembrane space.
Oh, I've been understanding mitochondria wrong my entire life.
It's a bit different. Yeah, it's a bit different. So there's like a compartment and then something like around that compartment creating another, like a second compartment, right? So there's like a space here in these like gaps and then a space inside of that also.
Wait, were you just saying that the inner membrane is what separates it, or is it the fold and stuff?
The inner membrane is folded. So this yellow is the inner membrane, and it is
Right.
folded, right? So the yellow is the inner membrane, it's super folded, and then all around that is the outer membrane, this covering.
Okay, I don't know why I asked that.
Does that make sense though?
Yep, it does.
Yeah. Okay. All right. Everybody breathe. Exhale. Get loose. Relax a little bit because we're about to do something that you're probably not going to enjoy. Okay. Not if, redox reactions, okay? Redox reactions are almost the basis of the rest of this respiration topic that we're gonna do, okay? Redox reactions are a combination of two reactions, a reduction and an oxidation. And I've always said to you that this is not a chemistry class, so I will try to keep it as straightforward and biological as possible, okay? Let me go back to the iPad for a second. That was supposed to be Jay's time to yawn. Okay. Okay. So redox reaction comes from reduction. That looks terrible. And oxidation. Now, if you look on the PPT, it says something like remember oil rig. Okay, I don't know oil rig. That's not how I learned it when I was growing up. I'm going to teach you what I learned. Okay, what I learned is Leo.
The lion says roar.
Leo the lion. Actually, no. Leo the lion. You could say Leo the saber if you wanted, but Leo the lion makes sense. Leo the lion says grr.
Roar.
Okay. Grr, right? Lion, grr.
Oh my god, IGCSE camps memories, no!
So what are we talking about here? Leo is, now this is backwards, so bear with me for a second. Leo stands for what? Uh-oh.
Lose electron.
Losing. Oh no, spelling is important. Losing electrons. Wow. Is
Oxidation.
oxidation.
Awesome.
Not awesome. Is oxidation and gain. What is G?
Gaining.
Damn it. Gaining electrons is reduction. Guys, I feel like I'm just like writing on a whiteboard. This is so cool.
Technology.
Okay. Gaining electrons is reduction. And I have way more colors than I would at school. Losing electrons is oxidation. Okay. I'm going to add one thing here, because whenever you lose an electron, you're essentially losing like let's say a positive and a negative. So sometimes we're going to see this as either an electron, which is, I will sometimes draw as like an E with a negative sign like this. Okay. But this could also be the loss of a hydrogen atom, often both. Okay, so oxidation is the loss of an electron or a hydrogen. Reduction is the gain of an electron or a hydrogen. We are going to see oxidation and reduction reactions happening throughout this process. Mainly, the one we're most interested in is, remember, the whole name of this is, what is it again?
Redox.
No, the fancy name for respiration?
Oxidative.
Oxidative, blah, 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 blah. So we're going to see a lot of oxidation happening to the glucose molecule. You'll see on the slides that they talk about oxygen also, like, so oxidation is also the gaining of oxygen, while reduction is the losing of oxygen. To keep it simple, just forget oxygen, because we probably won't see oxygen as part of this very often. Most of the time, we're going to be asking, did it lose electrons / hydrogen, or did it gain electrons / hydrogen? And that will tell you whether it was a reduction reaction or an oxidation reaction. Okay?
Wait, can you say the last part again? I didn't hear it.
So all I'm saying is when we look at like, actually, let me show you this, okay? So let's say, for example, random compound letter A, okay, becomes random compound letter A with a hydrogen, okay? Did A become oxidized or did it become reduced?
Oxidized.
Oxidized.
Okay, because it gained an electron / gained a hydrogen. So this was an oxidation.
Wait, what? No, you lose a electron.
Oh, sorry, not an oxidation.
No, oxidation is losing electron.
Wait, what?
Right, so it didn't lose, it gained.
What?
I know. Wait.
Yes, it gained electron. So that's why.
Yeah. No. My fault. Sorry.
Gain electron or hydrogen, right?
This is precisely the reason I quit chemistry.
Because with this, we assume that with this hydrogen comes one electron. Okay, let's go the other way around.
I'm confused. Like the hydrogen, like why is it? For bio purposes, let's pretend that hydrogen always comes connected with an electron. Let's just say that. Okay? Okay.
So if I say it loses a hydrogen, I'm also saying it loses an electron. If I say it gains an electron, I'm saying it also gained a
Then
hydrogen.
why is it plus and then minus?
Because a hydrogen
That's the
carries a
charge.
positive charge and an electron carries a negative charge. So
I'm a chem
together,
student.
they balance out.
Oh, so it's always like that, like even for reduction.
Wait.
Right.
Oxidation is gaining oxygen, so it's giving out electron. That's not reduction, it's oxidation.
Yeah, I'm gonna just tune out for this part.
I'm confused. Like, can you explain the minus and plus part again?
Yeah.
That's the charges.
Yeah,
Yeah.
but I don't understand.
Okay, so again, to not get, if we don't want to get to chemistry, just pretend, just assume that hydrogen and the electron always travel together.
Okay.
Okay. The reason I'm showing.
Minus.
Sorry.
Oh, that's just the like the bond thing.
No, but yes. Let's
I'm just trying to tell you that like sometimes they'll talk about this and sometimes they'll talk about this, but really the hydrogen and the electron, they're always like a combo.
Okay.
Okay. So if you're losing one, you're losing the other one also. If you're gaining one, you're gaining the other one also.
Okay.
Okay. Which is
It's
why
always
here I said losing electrons or losing H plus. This happens together, so that would be an oxidation.
Okay.
If you're gaining an electron, you're also going to be gaining a H plus. That's a reduction.
Okay.
Okay. So
Wait.
hydrogen atom and electron just comes as a combo deal.
Okay.
Okay. So if this compound B is losing an electron, then it is being
Wait, how is losing electron saying to having losing a hydrogen? Kind of lost.
don't worry about it. You can yeah. You can ask Mr. Oscar if you'd like, but for bio, if you start to think too much about that, you're gonna miss the important part. Like this is not that important.
Mr. Oscar is right.
The relationship between H plus and the electron is not important.
Okay.
Okay, all you need to know is it losing it or is it getting it?
Okay.
好的
Okay.
Okay, wait, just so to like clarify for like gaining hydrogen.
Wait, so the blue one is
gaining
gaining the electron.
Correct.
electron.
Oh, okay. Got it.
And then if you're losing hydrogen, you're also losing electron, and that's all you have to know. Okay.
Correct. Does that work?
Yeah.
Okay. So, two different reactions. Now, I just want to show you one additional thing that, let's say, if this part makes you confused, that's okay. Don't worry about it. But in chem, like people who are better at chem than I am, okay, you know that in order for A, in this case, for A to become A with the hydrogen electron, someone had to give them the hydrogen and electron, right? So something here, let's call them X, used to have this and then ended up not having it anymore, right? It's because this, actually, let's do it. Where did this come from? It came from some other compound, right? So in order for A to be reduced to A with this additional hydrogen or electron, it had to be reduced by something. In that process, X actually underwent an oxidation, right? So every time a redox reaction happens, that's why we call it redox, is because one of the things is getting reduced by something else and the other one got oxidized, right? Because X lost its electron. A gained an electron. So every time this happens, there's someone else involved in it. It doesn't just magically happen. So in this reaction also, something had to be there, let's call it Y, and then Y took away that atom.
Wait, Mr. Nilan?
Yeah.
Like, so whenever something oxidizes, something also like redox. No one's, I mean, like when something redox, something oxidizes.
There always has to be both happening.
Just two, okay.
Right? Because the thing that is being gained or lost has to come from somewhere, right? So there's actually like two reactions always happening. If this thing is gaining an electron, someone had to give it that electron.
Okay.
Because that thing gave the electron, that one lost one, right?
Okay.
Yeah.
Do
You
the-
cannot create
Correct.
a element.
Correct.
Wait, energy?
This is an energy, but sort of same kind of the same line,
Oh yeah, okay. Makes
right? So at the very basic point, you're just saying, okay, if this guy got an electron, who did they get it from? Oh, this other thing. If that one gave it away, it lost one, right? So one thing gets reduced, then the thing that helped to reduce became oxidized.
okay.
Is there anyone out there better at chem than I am that wants to add anything? Well, Ina, Amanda, I don't know who's good at chem.
Let me just turn up my volume for this one.
Good. I don't know who's like a chem star.
Chin.
Chin? We have special guest star Chin. Okay.
Hit him right now.
So I'm gonna switch back to the PPT.
Okay.
Yawn time. And okay. So that is actually like the reason I break this up is because this is something that always like gets people all riled up, gets your underwear in a knot, okay? And stresses you out. But really, keep it simple, right? Keep it simple. Are you losing or gaining electrons? So like hide one side of the screen, right? If it's losing, and again, I told you like ignore, just don't worry about this part for now, okay? Just forget about this, all right? Because these are the same. Is it losing an electron or is it losing a hydrogen? Then it's an oxidation. Is it gaining this? It's a reduction, okay? And instead of oil rig, you can remember Leo and Gur, Gur. Okay, so the last part is I'm gonna introduce you to two really important characters in this story, okay? The names of these characters are NAD plus, actually the important part is NADH and someone called FADH2. Sorry, let me get rid of this stuff. You're fine, chill. All you need to know are the letters, okay? These are called electron carriers. This term, electron carrier, where did it go? Where are so many things open here? Get out of the way. Okay.
By the way, Chin is here.
Oh, nice. Hi, Chin.
Hi, Mr. Nilan.
What's up? You missed my very probably terrible explanation about redox reactions. Yeah. We could have used you. Okay, this word, electron carrier, is very critical. So who are the electron carriers? The electron carriers are NADH and on the next slide you'll see F. Oh no, why did I choose to do this? I don't know. FADH2. Okay. What we will see is the kind of like the whole point up until the very last stage of respiration is to take electrons off of the glucose molecule so that we can transport them to the last step, okay? So all throughout this, let me show you this. What? All throughout this process. So from whether it's glycolysis, whether it's link, whether it's Krebs cycle, all these three things, what we're doing is we're kind of like, think of it like, you know, no, that's not really the analogy that I'm thinking of, but you know those like birds that sit on like water buffaloes or whatever, or those like big animals, and they like pick and like eat stuff off of them. It's kind of like that, where like the electron carrier is gonna like use glucose and just like steal electrons here and there, like all along the way, and with the express purpose of taking them to taking them to this last part. Oh, come on. The electron transport chain. So we're gonna like take electrons from here, and from here, and we're gonna bring them to the electron transport chain. But the thing, the vehicle that we use to transport them are these electron carrier molecules.
Wait, what are we talking about right now?
We are talking about how do we get the electrons to the electron transport chain. We use.
Which part of respiration are we talking about?
We will see this all the way through.
Shenanigans.
Right now, we're just naming them. We're just telling you the name, and I'm gonna stop. Okay. So in glycolysis, in the link reaction, in Krebs cycle, we're gonna see that we're going to basically make NADH so that it can go to the electron transport chain. We are going to make FADH2 so that it can go to the electron transport chain. How are we going to make it? Well, we're going to take FAD, which is nothing. It doesn't have electrons or doesn't carry electrons, and we are going to add electrons to it by reduction reaction, right? Gain electrons, and becomes FADH2, and then this will go to the electron transport chain eventually.
What's the difference between NADH and FADH2?
It's just two different molecules. A slight difference in their structure, but obviously FAD can carry 2 electrons, whereas NAD can only carry 1. No, sorry, there are 2 electrons here with an additional like hydrogen hanging out. So the whole reason why we learn redox reaction, not only what happens to the glucose molecule, is we're going to see the formation of all these electron carriers along the way, kind of like we make some in this process, we make some in this process, we make some, and then we'll see all of them will end up at this last stage and create a bunch of ATP.
They're all electron carriers, right?
Both, just these 2. There's only 2.
Okay.
Okay. And why do we need to know both sides of the reaction? Well, when they give away their electrons to the electron transport chain, then they are losing their electron, becoming FAD. Then it comes back to the start, gains electrons, goes there, gives them away, comes back, blah, 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 blah. Back and forth and back and forth.
Oh my God.
Oh my God.
Still very confused about why is the
I didn't hear anything just now.
Okay, I'll say this. That's where this first one of 5 ends. Okay, because if we keep going, it's way too much for one lesson. I wanted to just kind of set up the reactions, let you breathe, have the weekend, cry a little bit, and then be ready for Monday, where we actually get into things. We'll do glycolysis on Monday, so you'll see now how these reactions are really part of taking glucose and messing it up, so that we can get some energy out of it, and also just knowing the parts of the mitochondria. So I'll stop there, and now I am very, very open for questions.
We didn't learn anything about NADH and FADH.
No need.
Except they are electron carriers, right?
That's all you need to know.
Okay.
They can be reduced and they can be oxidized.
That's making me afraid of my university life.
Why?
There's a reason why I've invited the grade 12s to come join this topic over other topics. It's something you need to hear more than once, and like it's good to be refreshed about.
Now what?
Now what? Questions? I'm going to stop the recording so that this doesn't take 3 years to upload.